Okay, welcome back. So, in our series of histology lessons, we begin with this today under systems. So, I'm going to start with the systems histology. So, before we start, I'm going to request for two things. So, I'm going to say both things that are in this presentation and the things that are not in this presentation. So, people are supposed to pay attention to get both in order to not miss anything. Okay, so we start. So the respiratory system. Actually, before we go any further, the respiratory system, it's a system that you know is very cardinal for the survival of human beings. Okay, and other animals that need to respire. Okay, so this storage you are looking at is a storage of the human being. That's why we keep referring to the human being. So the first thing you have to know before we actually start is the gross anatomy of this uh, system. So it begins from the nostrils here, the nose, okay, up to the alveoli in the lungs. So number one, we have the nasal cavity. From the nasal cavity, we go um to the pharynx. To be specific, the nasopharynx. From the nasopharynx, we go to the rhinx. The rhinx, we come now to the trachea. Trachea of primary bronchus. Primary bronchi. From primary bronchi, we go to the secondary bronchi, which are intrapulmonary. From there, we go to respiratory bronchus, the terminal bronchus, to be very specific. Then the next we have the respiratory bronchus of the alveolar ducts. We have then the alveolar sacs, and finally the alveoli. So that is the outline of the respiratory system. So this respiratory system is divided anatomically into the lower and upper parts. Okay, it is divided anatomically into the lower and upper part. So the upper part ends just above the true vocal folds. So it is very important to emphasize that it ends just above the true vocal folds because we also have the false vocal folds. So it ends just above the true vocal folds. So it includes the nasal cavity, the paranasal air sinuses, the pharynx, and upper part of larynx. So most of this part is lined by the pseudo stratified columnar epithelium or the respiratory epithelium, which is ciliated. Okay, which is ciliated. So I say most of the um, I say most of this system is lined by ciliated stratified columnar epithelium with goblet cells. Okay, so we also have another division apart from the anatomical division. We also have the functional division, which divides this system into the conducting and the respiratory parts. Okay, yeah. So divided into the conducting and respiratory parts. So the conducting portion is from the nasal cavity and the terminal up to the terminal bronchioles. From the nasal cavity up to the terminal bronchioles. That is the conducting part. Okay, so it is very important to understand this system in this uh, uh, case, or it's very important to understand these kinds of divisions. You must be able to comfortably uh, define the boundaries in terms of anatomical division and functional division. Okay, so the conducting portion of this system is from the nasal cavity, the nose here, up to the uh, terminal bronchioles. So from the nasal cavity up to the terminal bronchioles, that is our conducting part in terms of the functional division. That is our conducting part. Then from the level of the respiratory bronchioles up to the alveoli, that is the respiratory part of this system. Okay. So now we're going to specifically start looking at the exact parts in order from the beginning up to the end. Okay, so we're going to start with the nasal cavity. So from here, the nasal cavity is also divided into two parts. We have the anterior dilated part or the external dilated part, which is called the vestibule, and the internal nasal cavity. Okay, so if you can palpate your nose, you can actually feel that the anterior part of your nose is made of soft tissue, and as you palpate upwards towards the bridge of the nose, you can feel some bones there. So those bones, inside those bones, there's a cavity. So that is what we call now the internal nasal cavity of the vestibule. So that those are the two divisions of the nasal cavity: the vestibule and the nasal cavity. I mean the internal nasal cavity. Okay. So the skin of the nasal cavity is in the vestibule has these important things here to take note of. So number one, understand that the skin in the vestibule of the nose is actually coming from outside. So as it goes into the vestibule, it is lined by the same epithelium that lines the external surface of the skin. Okay, that lines the external surface skin. Okay, so in the vestibule, you get to find these three important things that you must understand and never forget. We have the sweaty glands, the sebaceous glands, and we have hair, which is we have this coarse hair which we call the vibrissae. So these are to be remembered. Most questions come here, they'll just say which of the following is not found in the vestibule of the nose. Then now they'll put these very options and put something else there. And if you do not know, you obviously get it wrong. Okay. So just to emphasize, note that in the vestibule, the epithelium changes from a keratinized stratified squamous epithelium to a typical pseudostratified columnar epithelium with a transitional zone of stratified squamous non-keratinized. So in the vestibule also there's this one important thing that happens. There's a change of epithelium, transition. Epithelium is transitioning from stratified squamous keratinized epithelium to the respiratory epithelium, which is pseudostratified columnar epithelium with goblet cells. So in between, in the transitional zone, we find now the stratified squamous epithelium, which is non-keratinized. So we're actually moving from stratified squamous keratinized, stratified squamous non-keratinized, then to pseudostratified. That happens in the vestibule of the nose. Okay. Yeah, I hope we get it that there. There are no questions. So then we go to the respiratory epithelium now. The respiratory epithelium. So this respiratory epithelium is also called, or it is seriated pseudostratified columnar epithelium with the goblet cells. So don't forget. So if a question comes, especially in projections, the practical exams, if you can't write this, all the details that are appearing here in red, just simply write this one, respiratory epithelium, so that you don't lose any marks. Because if you decide to write this and leave any uh, word here, then it means you are going to lose some marks. So to avoid losing marks in such a manner, just write this one if you have an opportunity to. Okay. So in this respiratory epithelium, we find five major types of cells. That is what is very important. That is what is very important. So we find five major types of cells. So these cells, we're going to look at them one by one. So note also that all these cells that are found in this epithelium touch the basement membrane, and this basement membrane is unusually thick. So compared to the actual epithelium, this basement membrane is thicker than the epithelium. Okay, basement membrane is thicker compared to the epithelium. Okay, so yeah, so now you can compare this epithelium now we're talking about to the olfactory epithelium, which we're going to look at later. Okay, so let us now go into cells. So of the ciliated columnar cells, so these ciliated columnar cells, as the name suggests, they have cilia on the apical surface and are the most numerous of the 5 types of cells found in this epithelium. Okay, so that is all you have to know about these ciliated cells. That they are ciliated, they have cilia on the apical surface. So the cilia is not on the basal surface. So pay attention to where the cilia is. It's on the apical surface, not on the basal surface. Okay, yeah. So the other type of cells we find there are the goblet cells. So the goblet cells, these are also numerous, uh, but not as numerous as the serrated columnar cells. But in some areas, you find that these cells actually predominate. Okay, they have basal nuclei. They have basal nuclei. So these goblet cells, their function is to produce mucus. So in other words, they're mucus producing cells. Okay, so their nuclei is at the base. So compared to astnous cells, these have basal nuclei, but the astnous cells have apical or central nuclei. They actually have central nuclei. Yeah. So these goblet cells, in their apical domains, they are filled with granules. They are filled with granules of mucin glycoproteins. So these mucin glycoproteins, once they get mixed with this, water or other fluids, that's when now they form mucus, which is yeah, then can accumulate in the airway in some diseases. Okay, okay. So this mucus, once produced, it functions to trap unwanted materials. Okay, like pathogens, dust particles, and other particles that may go with air into the airway. So once they're trapped in this mucus will now be swept out of the airways and to the outside. Okay. So the third type of cell that you find in this epithelium are the brush cells. So the brush cells, as their name suggests, so brush, it means they have some microvilli on the surface. So in anatomy, most times when you hear this word brush, it is formed by microvilli. I'm sure you've heard of brush border that you can find in the proximal convoluted tubules of the uh, nephron and yeah, obviously in other areas. Yeah. So these brush cells, they are much less numerous compared to the above two cells, the goblet cells and the ciliated cells. So these ones are less numerous. Okay, they are less numerous. So in terms of shape, these cells are actually columnar in shape. They are columnar in shape, just like the ciliated cells. These brush cells are columnar in shape. So they have microvilli on the surface. They have microvilli on the surface. So here, said compare with columnar cells above. So the columnar cells, instead of having microvilli, they have cilia instead of microvilli. So be able to differentiate and be able to know the salient features of these cells. Okay. The next type of cells, in fact, before we move, so these cells are chemosensory cells. So they're chemosensory cells. So that means they're able to sense some chemicals. Uh, they're able to sense some chemicals. Okay. They're chemosensory receptors. They receive, okay, sensation of such chemicals. Okay. So these cells also resemble gustatory cells. So these gustatory cells are actually found uh, on the tongue. especially associated with the papillary. The taste buds are found in the taste buds, to be very specific. So these cells, they are they both function to uh, transfer signals, or what you call signal transduction. Okay, so that is about the brush cells. That is all you have to know about the brush cells. So the next type of cells we're going to look at are the small granule cells, the small granule cells, or the Kuchsky cells, small granule cells or the Kuchsky cells. So these cells, just from their name, granule cells, they have they have a core, or they possess what you call a core of granules. So it is from this core of granules that they derive their names, small granule cells. So these granules are very small, that's why called this name. So these are the granule cells. So these granule cells belong to what we call the diffuse neuroendocrine system. So that means they're actually endocrine cells. They're endocrine cells. I don't know which hormones they produce, but just know that they are endocrine cells. Okay. So they are of the same population as the brush cells. So these two cells, the brush cells and the small granule cells, each of them comprise of 3% of all the cells that are found in the respiratory epithelium. Then we have the basal cells. The basal cells, these ones now are stem cells. They are stem cells which produce the other types of cells. Okay, the that is about the respiratory epithelium. So what I'm emphasizing here is know all these types of cells and their properties. Okay, that is in terms of structure and function. Okay, know these cells. Then one thing you must note now here is that the respiratory epithelium, the other thing of importance that you have to know, because at the end of it all, you must be able to differentiate this epithelium from the olfactory epithelium. So these two epithelia must be treated like twins that are identical, and you want to differentiate them so that you stop confusing their names. Okay, so you have to pay extra attention to the salient features for each of these uh, epithelium. Okay, so respiratory epithelium has so in comparison to the olfactory epithelium, the respiratory epithelium has a thinner epithelium itself compared to the olfactory epithelium, but the basement membrane of this epithelium is very thick compared to the basement membrane of the olfactory epithelium. Okay, so now we go to the next. We now look at the olfactory epithelium. Okay, so the olfactory epithelium is found only in two places in the body. So it is found only in two places in the body. So it is found in the roof of of the nose, and also the superior nasal concha, or which you can also call the turbinate bone. Okay, it's found only in the roof of the nose and the superior nasal concha, which you can also call the turbinate bone. Okay, so these turbinate bones. So in the nasal cavity, we have where can I get take a thing? Okay, I wanted to draw something, but then I can't find the yes. But anyway, so on the laterals of this nasal cavity, on the laterals of the internal nasal cavity. So from each lateral projects three bones. Okay, three bones that form structure like shelves. I'm sure you know shelves, isn't it? Yeah. So from each lateral projects these three bones. So these three bones, in order from above downwards, we have the superior, middle, and inferior nasal concha. So I'm saying on the superior nasal concha is lined by this olfactory epithelium. Apart from the superior nasal concha, the other part that is lined by this olfactory epithelium is the roof of the nose. Okay. Okay. So this point here I've always emphasized is very important. So note that the respiratory epithelium has a thick basement membrane and a thin epithelium itself. So as opposed to the respiratory epithelium we looked at recently. The olfactory epithelium has three types of cells, three main types of cells instead of five. This one has three. So compare with the respiratory epithelium. So you can compare these cells as we talk about them. You compare the features they have to those cells that we talked about in the respiratory epithelium, and also compare the epithelium itself in terms of other features. Okay. So the olfactory neurons, the first type of cell that we're going to find in here and that we're going to talk about here. So note that this olfactory neuron is actually a bipolar neuron. So when you look at nervous system, you will learn these types of neurons. Okay. You look at these types of neurons. So the bipolar neurons are found. Also, I'll give you three sites where you can find these bipolar neurons. So bi means two. So bipolar neurons. So as a mnemonic, to remember where these bipolar neurons are, you have to remember the parts of your body that are actually paired. So number one, you have the eyes. So your eyes are paired. So in the retina, you find these bipolar neurons. So you have your ears. Your ears are paired. So bi for two. Your ears are paired. You are going to actually find these bipolar neurons in the internal ear. Okay. Then that site is here. Is here. The nose. This one is a bit trick. How is the nose paired? Okay, so I'm going to say that the nose has got two openings, what you call the nostrils. So that is how you're going to remember that. You are going to find bipolar neurons here because of these two nostrils. Okay. Yes. So one thing you must note about these olfactory neurons is that they their nuclei form an irregular row near middle of epithelium. So they form like, so their nuclei are concentrated in the middle of the epithelium. So such a way that it form an irregular row like that. Okay. So on their apical surfaces, these olfactory neurons have got dendrites and a knob-like swelling with a dozen of basal bodies. So from this dozen of basal bodies emanate what we call cilia. So cilia always emanates from basal bodies. Okay, cilia always emanates from basal bodies. So from this dozen of basal bodies emanate cilia. So this cilia that emanates from these basal bodies is non-motile. So this is non-motile cilia. That's why in some past papers you find the question that to say which of the following is true. Okay, the question may come like that. Which of the following is true? Okay, then they will say cilia found in the ear is immotile. Okay, or cilia that is found here in the olfactory epithelium is immotile. So that is true because of this point here. Okay, that is true. Okay, so now these dendrites that are actually coming from these bipolar neurons will move, will come together and form what we call the olfactory fila. This olfactory fila will actually send axons that will pass through the cribriform foramina in the ethmoid bone. You are going to understand more of that when you now progress to third year and start looking at head and neck anatomy, gross anatomy. Okay. Mhm. So the second type of cells that we're going to find in this epithelia are the support cells. The support cells or the sustentacular cells. Support cells that can also be called sustentacular cells. So these cells are columnar in shape with narrow bases and cylindrical apices. Okay. Have apical microvilli and nuclei. Have apical microvilli and nuclei. So now compare these cells. Compare these cells with the those found in the respiratory epithelium. So which cells in the respiratory epithelium did we say have cilia on the have macrovilli on their apical surfaces? So if you paid attention, you realize that those are the brushy cells. So the brush cells and the support cells in this epithelium have got apical microvilli. Okay. So one thing to note also is that these support cells are linked to the olfactory neurons via junctional complexes. Via junctional complexes, okay? So the last type of cells that we're going to find in this olfactory epithelium are the basal cells. So the basal cells, in terms of shape, they are small, spherical or cone-shaped cells. Okay, these cells, as usual, they are stem cells replacing other two types of cells, which are the support cells and the olfactory neurons. So olfactory neurons are actually replaced more frequently than the support cells. Uh, so they're being replaced every 2 to 3 months. They're being replaced. Okay. So the lamina propria has large serous glands. So the lamina propria underlying this olfactory epithelium has serous glands, which you call the serous glands of Bowman, or you can simply call them Bowman's glands. So this is very important. These glands are very important, just like von Ebiner's glands in the tongue. These glands are very important because they produce this serous fluid. So this serous fluid, once it is produced, it actually cleans these olfactory neurons, because these olfactory neurons, they have to sense smell. So when smell particles bind to these neurons, that these neurons are going to detect what type of smell it is. Okay, so if these neurons are not going to be uh, to be cleaned by this serous fluid, it means if we if this type of smell particles bind on there and they stay there, to me the next type of smell that is going to need to be sensed or bind there will not be recognized, because there will be already something that is already there. So that is why it is important that this fluid, these glands are there, and they produce this fluid so that they keep cleaning those neurons, so that they keep binding new uh, uh, odoriferous elements. Okay, that is why these glands of Bowman are very important. So always remember that these glands are found where. So the location of these glands, don't just know that these Bowman's glands are there. You must also be able to give their specific location. Okay, so in this case, they're found in the lamina propria. They're found in the lamina propria. Okay. Yeah, so that was about it, the two types of epithelia. So if you understand now those, we can now proceed to looking at the specific parts. Okay. So also of the paranasal air sinuses. So these paranasal air sinuses are openings in bones. They are openings in bones. So some of the bones that have these openings are the maxilla, ethmoid, sphenoid, and frontal bones. So you can remember these by using this mnemonic key, MESI for here. Okay, you can remember these bones. There might be other bones that have, but these are the ones that have been mentioned mostly in literature. Okay, so why are these paranasal air sinuses there? Why is it necessary that we must have these paranasal air sinuses? Okay, so these actually function to reduce head weight. Okay, these function to reduce head weight. Without these air sinuses, these bones were going to be very heavy, and as such, maybe the neck would have even be failing to support the head because of the heaviness of these bones. So these bones are very heavy. So to reduce some of their weight, they are perforated. They have these holes inside, which you call the sinuses now. Okay. So I'm sure some of you have experienced this when actually maybe you have uh, flu. When one has flu, okay, what you call rhinitis, okay, you find that these paranasal air sinuses are blocked. Mucus collects in them. So you find that you start feeling as if your head is heavy. Okay, you start feeling as if your head is heavy. So that is because these air sinuses are blocked. Okay, that is because these air sinuses are blocked. Okay, so these are important things that you have to know about these paranasal air sinuses. So number one, these paranasal air sinuses are lined by respiratory epithelium. So the same epithelium that lines all other parts of the respiratory system It's the same epithelium that lines these paranasal air sinuses. Okay, so this is uh, a very important point that most examiners target. They'll just modify this statement and let you be able to figure it out. Okay, so always remember that these paranasal air sinuses are also lined by the same respiratory epithelium, which what we call ciliated pseudostratified columnar epithelium with the goblet cells. So even goblet cells are there in this in the lining of these paranasal air sinuses. Okay. So lamina propria has fewer glands and is continuous with underlying periosteum. So periosteum, this is the outer covering of uh, compact bones. Okay, outer covering of bones, that is periosteum. So this lamina propria that lines, because these cavities these sinuses are inside the bone, so the lamina propria of their epithelia is continuous with the periosteum that line those cavities. Okay, so also note that these paranasal air sinuses communicate with the nasal cavities. They communicate with the nasal cavities. So most examiners will bring this part here in a question, then put it as an option. Then they'll say these paranasal air sinuses do not communicate with nasal cavities. So you have to pay extra attention. Pay attention to every detail and every line. Okay. Okay. So just a clinical correlate. So inflammation of these air sinuses is what we call sinusitis. Sinusitis here. Okay. So trachea now. Come to the trachea. So the trachea, in terms of length, it is about 10 to 12 centimeters. It begins at C6 vertebral level. And these are some of the important features that you have to know. So that was part of gross anatomy. But now we're looking at histology. So this trachea has got what we call seromucous glands uh, in the submucosa. Okay, submucosa here, pay attention, not mucosa, the submucosa. So these seromucous glands produce watery mucus. So the importance of this mucus, just like we said earlier, is to trap any unwanted materials that are coming inside the airway with the air. Okay. So in terms of support, the trachea is supported by C-shaped hairline cartilage. C-shaped hairline cartilage. So pay attention to what type of cartilage. So the type of cartilage here is we're saying it's hairline cartilage. Then what shape is this hairline cartilage? It is C-shaped. Okay, it is C-shaped. Okay, so this it is C-shaped. So meaning one side is deficient. Okay, so the side that is deficient is posterior side. So on the posterior side, this hairline cartilage is deficient, and this is where it is opposed. The trachea is opposed to the esophagus. So why is it important that this cartilage must be deficient posterior? So this is to allow the esophagus, since the esophagus is behind the trachea, just behind the trachea. So that when you swallow something, so the esophagus is almost always collapsed. So when you swallow something, okay, you swallow something, the esophagus will have to expand as that bolus of whatever you swallowed is moving down. So that deficiency is to allow for the esophagus, the collapsed esophagus, to expand whenever you swallow something. Okay, that is why it is important that this cartilage is deficient posteriorly. So always remember that the cartilage is deficient posteriorly. Okay. So the next point says deficient part. So this deficient part is not completely deficient, like as in just a gap without anything. No. So there's this type of muscle which we call the trachearis muscle. So trachearis muscle is not only enough to know that behind the esophagus, what completes the deficient part is trachearis muscle. You also have to know what type of a muscle it is. So it is a smooth muscle. Okay. So from there, also remember that this hyaline cartilage that supports the trachea has a perichondrium. Has a perichondrium. So if you did basic histology of cartilage, you actually remember that the only type of cartilage that does not have perichondrium is um, the articular cartilage, which is hairline, and also the fibrocartilage, which actually depends on the perichondrium because it is always neighbored by hairline cartilage. So for nutritional needs, it depends on hairline cartilage. So this hairline cartilage, remember that it has its perichondrium, which is the outer covering of uh, uh, this cartilage. So there we said the periosteum, here we are saying perichondrium. So these are two different things. Periosteum is the outer covering of a bone. Perichondrium is the outer covering of cartilage. Okay. So again, it is not just important to know that. It is not only enough to know that there is this hyaline cartilage for support. You also have to be able to give precise location in the wall of the trachea of this cartilage. Okay, so it is in between the submucosa and adventitia. Okay, it is in between submucosa and adventitia. Other books will say it is just within the submucosa, but it is not in adventitia or submucosa. It is in between these two. Actually, if we are to be very specific, we can actually say it is the one that separates the submucosa from adventitia. Okay, so glands that are mainly found in the trachea are mucus glands. So this is statement here, mainly found. So that means we can also find serous glands, but the glands that dominate there are the mucus glands. Okay, so that was all we needed to know about uh, trachea. For further reading, you can actually consult your notes and other sources of information. So now this brings us to the bronchi. So the bronchi, these ones we have two types of the primary bronchi and the secondary bronchi. So the primary bronchi are the ones actually uh, that are given off when trachea bifurcates at the T4, T5 vertebral level. Okay, so it bifurcates into left and the right primary bronchi, of which the right side of the primary bronchi is actually more vertical compared to the left, and that is important clinically because it implies that when something foreign goes into the nostril, it will mostly follow the vertical part. It will mostly launch itself into the right primary bronchi and actually get into the right lung, okay, and into the uh, lower uh, lobe, okay. Yeah, let me not give more information that will start to confusing in the end. Okay, so now histologically we are saying these bronchi are actually uh, similar uh, to the histology of the trachea. That's why in most books you'll never find uh, them talk about them. Okay, but it is important that we enlighten ourselves of some of the features that these bronchi possess. Okay, so number one, so we're saying this bronchi are actually similar to trachea except for two important things. So here, number one is organization of cartilage. So organization of cartilage. So in trachea, we said that cartilage is C-shaped and is deficient posteriorly. Here we are saying, of course, we find the C-shaped type cartilage in the bronchi, but most of them are complete. So meaning they form a ring. So you've seen the importance in primary bronchi, we can form a complete ring of this hyaline cartilage because there is nothing that we can actually disturb behind. So in terms of trachea, we can disturb the esophagus if you are to put a complete ring of this hyaline cartilage. So we must be able to relate things like that. Okay. So the second thing is arrangement of smooth muscle. So the smooth muscles arranged here. So in the trachea, these smooth muscles are arranged in terms of trachearis muscle behind, isn't it? Behind. But here we find that this smooth muscle is actually in lamina propria and it crisscrosses in bundles and is arranged. Spirally arranged like that. So this way here. Okay. So the other thing we get to find in the bronchi are the lymphatic nodules. So lymphatic nodules are present in the bronchi. Okay, especially at branching points, you find a lot of these lymphatic nodules. These these lymphatic nodules are meant for immunological defense or immune responses. Okay, so as bronchi become smaller, so pay attention to this statement. As these bronchi become smaller, there are these things that happen. Number one, smooth muscle elastic fibers, and what we call the mucosa associated lymphoid tissue mouth become abundant. Pay attention to this word. What happens to these smooth muscle, elastic fibers, and the mucosa associated lymphoid tissue become abundant or increase in amount or numbers. Okay. Then cartilage and other connective tissue reduce. Cartilage and other connective tissue reduce. So pay attention to this. Smooth muscle, elastic fibers, mouth, they increase as you go down uh, uh, res- uh, the respiratory tree. Then cartilage and other connective tissues reduce. Okay. Okay. So note also that the trachea and the bronchi are aligned by respiratory epithelium. Aligned by respiratory epithelium. Okay. Pay attention to that. Next, we go to the terminal bronchus. Next, we go to the terminal bronchus. So just like for the primary bronchi, we said we had secondary and primary bronchi, of which the secondary bronchi were actually found intrapulmonary, meaning inside the lung tissue, inside the lung tissue. So these bronchi actually inside the lung tissue, as they go, they will divide and give us the terminal bronchus now. So the terminal bronchus are intralobular airways, are intralobular airways, intralobular airways, of which now we can say the secondary bronchi were actually interlobular. So intra means inside the lobules, then interlobular means in between lobules. Okay, that was it. The secondary bronchi were in between lobules, but now these terminal bronchioles are intralobular. So meaning they are inside the lobules of the lung. Okay, so they are formed after about 10th generation. So this one you understand, as especially in physiology, there is actually a stepwise a division of these things. They divide. We call them generations. So after each generation, there are some changes. So you can, if you are more interested to know, uh, you can actually find. You can actually look up that information. Okay, but what is important here to know is that these are formed after about 10th generation. Okay, so this here is also very, very important. That's why it is highlighted in red. Okay. So these terminal bronchioles do not have submucosal glands or cartilage. So we said the cartilage, as you go down, it reduces. So I've actually confirmed that this is true because these terminal bronchioles do not have cartilage. Do not have cartilage. Neither do they have submucosal glands. Okay. So in larger bronchioles, epithelium is respiratory, though low in height and complexity. So the other thing we have to take note is that as we go down the respiratory tree, the epithelium was changing. So initially we had the stratified squamous epithelium in the vestibule, changed to um, pseudostratified or respiratory epithelium. So respiratory epithelium at some point, so that is now when you reach the level of terminal bronchioles, it is actually changing now. It's actually changing. So it is becoming uh, shorter and shorter because in the by the time we reach the alveoli, the epithelium must be very simple, must be simple squamous. That is to facilitate diffusion or exchange of gases there. That is why the epithelium must be very thin to reduce diffusing distance, to reduce diffusion distance. Okay, yeah, okay. So one thing also you have to note here that the mucociliary apparatus begins in the terminal bronchioles. So this is a system that ensures that the mucus that has trapped dirty and other particles is actually swept out of the airway. Because if it is allowed to stay in the airway, it will eventually block the airway, and that will lead into suffocation. Okay, so this mucociliary apparatus is actually cilia that is sweeping. So cilia is there in the airway to sweep this mucus out of the airway. So that sweeping process begins in the terminal bronchiole. So that means the mucus that has collected in the terminal bronchioles will be swept upwards into the secondary bronchi. From the secondary bronchi to be swept up now to the mm, yeah, to the primary bronchi. From the primary bronchi to be swept up into the trachea, all the way up until it is uh, out of the airway. Okay, so the cuboid epithelium of terminal bronchioles. So the cuboid epithelium. So you can see the epithelium is keeps on changing. So the cuboid epithelium of terminal bronchioles consists largely of club cells. So these club cells can also be called the bronchiolar exocrine cells, or you can also call them Clara cells. Okay, so these are very important cells, as we can see, as we are going to see from their functions. So now we're going to look at these cells in detail. So number one thing you have to note about them is that they are non-ciliated. So the only cells that are ciliated here, well, we've already mentioned, and we know the cells that are ciliated and those which are not ciliated. Okay, okay, yeah. So I've actually mentioned that olfactory neurons are ciliated. Okay, so these Clara cells are non-ciliated. Okay, in terms of shape, they are dome shaped. They're dome shaped. So that is what you have to know, those specific features. So they're non-ciliated and they're dome shaped. So that now brings us to their functions. What are their functions? So these Clara cells secrete what we call surfactant like proteins. Surfactant like proteins. This one is highlighted in red. So very, very important. Very, very important to take note of the difference. Okay, take note of the difference. These Clara cells do not produce surfactant. They do not produce surfactant, but they produce surfactant like proteins. So these proteins are actually lipoproteins. So sometimes they produce surfactant lipoproteins. Okay, so they are, what is important is that those things are what are actually proteins. So they produce surfactant like proteins and mucins. So take note, they do not produce surfactant. So this is one confusion that actually examiners want to write down so that to confuse their students. So they'll put this actually, this is a very option, and they'll say Clara cells produce surfactant. That is very, very false because they do not produce surfactant. Instead, they produce surfactant like proteins or surfactant lipoproteins, okay, and mucins. Again, here they produce mucins, not mucus. So these mucins, once they are produced and they mix with water, that's when now they become mucus. So these Clara cells do not produce mucus, but they produce mucins. Okay. So the other function of these cells is that they are actually de- uh, detoxify inhaled xenobiotic compounds. They detoxify inhaled xenobiotic compounds. So this is your homework now. Go and find out what the xenobiotic compounds are. Okay. So secretion of antibacterial peptides and cytokines for local immune defense is another function of these grass cells. So they secrete antimicrobial peptides and cytokines. So cytokines, these are communication molecules. Then antimicrobial peptides, so mean these are peptides that are just work against microbes. Okay, so that is for immune defense or local immune defense. Okay, that is within the airway. That is local immune defense. So these peptides that are being produced in the nose, they will not go and function in the GIT. That is why we say they produce these secretions for local immune defense. Okay, just for defense in the respiratory tract. Okay, yeah. So take note also that brush cells and Kuchinsky cells are present in the terminal bronchioles. So apart from the Clara cells, we also find these two types of cells, the Kuchinsky cells and the brush cells, which we found already and explained about them in when we're looking at the respiratory epithelium. So we also find them here in the terminal bronchioles. Okay. So smooth muscles and elastic fibers are present in lamina propria. So note here that in the terminal bronchioles, we find smooth muscle and elastic fibers. As we said, going down the respiratory tree, these smooth muscle and elastic fibers increase. Okay, so we find these to be very specific. We find them in the lamina propria of these terminal bronchioles. So you always have to remember to be specific in anatomy. You always have to remember to be specific. So these are found in the lamina propria. Okay, okay. So I wasn't adding slides for to show the structures of these things. What I was interested in is uh, theory, not a practical. So if you actually get a histological slide of these terminal bronchioles, you are going to see that their epithelium uh, is actually folded. Is actually folded. So that folded appearance is actually due to underlying smooth muscles in the lamina propria. When they contract, they squeeze that epithelium and it forms into those foldings you see. Okay, yeah. So compared now to the bronchi, these bronchioles or the terminal bronchioles have more smooth muscle than the bronchi. Okay, so that now brings us to the respiratory bronchioles. So note that respiratory bronchioles and terminal bronchioles are both the bronchioles. Then where is the difference? So the difference is the major difference that respiratory bronchioles actually have alveoli in them. So therefore, these respiratory bronchioles are able to do some form of respiration, and that is why they are called the respiratory bronchioles, because they are able to do some kind of respiration because they contain these sac-like alveoli. Okay, they do not contain actual alveoli, but they contain sac-like alveoli. That is very important to understand. So these sac-like alveoli open to the lumen of these respiratory bronchioles via what you call the alveolar openings. Via what you call the alveolar openings. Okay, so club cells are also present in the respiratory bronchioles. So in general, we can say these club cells, which you also call the bronchiolar exocrine cells or the Clara cells, are only found in the terminal bronchioles. So in the bronchi, you're not going to find these cells. You're not going to find these cells in the alveoli. No, they are found in the bronchioles. Okay. So elastic fibers and smooth muscle are in lamina propria. So you should now be ringing in your head to say these actually elastic fibers. Always expect to find them in the lamina propria. Always expect to find them in the lamina propria. Okay. So simple squamous cells at alveolar openings extending into alveoli. So these alveolar openings are actually lined by simple squamous cells that also extend into these alveolar sacs. Okay, that is all about respiratory bronchioles. So next we go to the alveolar ducts. So alveolar ducts, so the respiratory bronchioles now further divide and give us now the alveolar ducts. So these alveolar ducts, they are almost entirely lined by openings of alveoli. Because as we go down now, alveoli is increasing. Alveoli is increasing. So it means in the alveolar ducts, we are going to have many openings of these alveoli, such that the same openings will now actually become the lining of these alveolar ducts. That is very important to take note. Okay. So we also have strands of smooth muscle surrounding these alveolar openings. They surround these alveolar openings, these smooth muscles. I don't know if it's to control the opening or what, but just know that they surround these alveolar openings. Okay. So these alveolar ducts end in what you call alveolar sacs, which are clusters of alveoli, So also elastic and collagen fibers support both the duct and its alveoli. So both the duct and the alveoli are supported by elastic and collagen fibers. Okay. So I've already mentioned this point. So perhaps to emphasize on one thing. So clusters of alveolar sacs occur occasionally distally along length of alveolar ducts. Okay, so these alveolar ducts are not uh, so very many in this alveolar duct, but they occur occasionally uh, at irregular intervals, especially distally along the length of these alveolar ducts. Okay, so epithelium is now essentially thin. That is one thing we also have to note. So he said by the time we reach the alveoli, the epithelium must be simple squamous. Okay, the epithelium must be simple squamous. So we're seeing that as we are going down the respiratory tree, the epithelium is changing, and at this level of alveolar ducts now, it is essentially thin, consisting of only a web of elastic and reticular fibers, which surround the alveolar openings and alveoli. Okay, so capillaries are also closely applied to them. So now, remember, this is now the functional part of the lung. Now, this is where now respiration is taking place. So respiration, gaseous section, this one is blood that is coming to the lung should be oxygenated, and the carbon dioxide that is in that blood must be taken out. So that is why it is now important that these blood vessels in form of capillaries now come to lie close to these alveoli here. Okay, that is why it is important. So that is now to initiate the process of gaseous exchange that is occurring in the lungs. Okay, so we now come to the alveoli. Now come to the alveoli. So the alveoli are the functional unit of the lung. They are the functional unit of the lung. So note that uh, at the level of the alveoli now, this epithelium is now simple squamous, lined by two types of cells. So these two types of cells are what we call type 1 pneumocytes or type 1 alveolar cells, and type 2 pneumocytes or type 2 alveolar cells. Okay. So now here is a very confusing point. Here is a very confusing point, and pay attention. Pay attention. Most students of anatomy get confused here. So pay attention so that you not become a victim also. Okay. So we're saying that this alveolar lining, or the lining of the alveoli, is made up of two types of cells, okay, which are the type 1 and type 2 pneumocytes. So now the lining, 95%, we're saying 95% of the lining of the alveoli, or maybe before I come to that point, not that uh, both types of cells, type 1 and type 2 pneumocytes, lie in the alveoli. Okay? Both types of cells, type 1 and type 2 pneumocytes, lie in the alveoli. What differs is the percentage of the alveoli they line. So I'm saying 95% of the alveoli is lined by type 1 pneumocytes, and then 5% of each alveoli is lined by type 2 pneumocytes or the alveolar cells. So take note, this is one point. The other point is here. The other point is here. So in terms of population, we are saying type 2 pneumocytes are more than type 1 pneumocytes. So you see why I'm saying it's confusing. So in the lining of the alveoli, in terms of population, type 2 pneumocytes are more than type 1 pneumocytes. But type 1 pneumocytes line uh, a larger surface of the alveoli compared to type 2 pneumocytes. How? So it's very confusing. So just know that type 2 pneumocytes, in terms of population, are more than type 1. But in terms of lining of the alveoli, type 1 pneumocytes line 95% of this alveoli, and type 2 pneumocytes line only 5%. Okay? So 60% to 40%, that is a division of which we have already talked about. So take note of these 2 points because they are confusing, and since they are confusing, they become the target of examiners. They become the target of examiners. Okay? So, a little more specific now to the cells. So, type 1 pneumocytes are squamous in shape, while type 2 are cuboid in shape. Those are differences now. But despite these differences, these cells have the same origin. They have the same origin embryologically. You understand that when you do embryology. Okay. So, type 2 pneumocytes are also called septocells, and they have lamellar bodies. They have lamellar bodies in their cytoplasm. So, the neighboring alveolar cells. So these alveoli now, in fact, here it's not supposed to be alveolar cells. So we're saying neighboring alveoli. So please take note of this. You can just cancel this one, alveolar cells. It's not alveolar cells, it's neighboring alveoli. Okay? So neighboring alveoli are opposed onto each other, and the touching surfaces of these alveoli form what you call the alveolar septum. So we have one alveoli here, another alveoli here, then there's this part here where these two alveoli attach. This part is what you call now the alveolar septum. This part where the two neighboring alveoli are attached to each other is what we call the alveolar septum. So now this alveolar septum has got some important things in it that you have to take note, and you have to know them by heart. Even when someone wakes you up from sleeping and ask you that question, you must be able to enlist these things that are found in the alveolar septum. So without wasting much time, let us look at them. So number one, we have fibroblasts. So fibroblasts, these are connective tissue cells. Their function is to produce extracellular matrix, and hence, where we have fibroblasts, we are sure of having extracellular matrix. So therefore, the second thing that we find in the alveolar septum are the uh, is the extracellular matrix. I mean, so this extracellular matrix is actually sparse and it's made of elastic and reticular fibers. It's made of elastic and reticular fibers. Okay. So the reticular fibers prevent collapse and excessive distension of these alveoli, whereas elastic fibers allow these alveoli to distend when filled with air. Okay. So the other thing we find there are the macrophages and the leukocytes. So we've always known that these macrophages belong to what we call the mononuclear phagocytic system, and they come from monocytes. So wherever these monocytes shift to, they will assume a different name. So in this case, the monocytes that shift to the lungs are called now the dusty cells. So instead of saying macrophages, you can be very specific and say dusty cells are found in the alveolar septum. So the other thing that we find in the alveolar septum are the capillaries. And why should there be capillaries? Capillaries are now getting so close to the alveoli to enable or facilitate gaseous exchange. And that is why you find these capillaries in there. Okay. So other thing we find there are the alveolar pores of cone. So in the alveolar so we're saying the alveolar septum is actually perforated. So it has got these holes which you call pores. So these holes or pores which are called alveolar pores of cone, they connect two neighboring alveoli. So where two neighboring alveoli actually oppose and are connected to each other is what we call now the alveolar septum. So this alveolar septum is actually perforated. It has got holes that are coming from one alveoli to another. So why are these pores of cone, alveolar pores of cone, important? So each alveoli has got a mechanism or has got a duct that fills it with air. So in the case of that duct that fills one alveoli with air is blocked, so it means the duct that fills the other alveoli is able to fill the alveoli it is connected to, and once that alveoli is filled, air should be able to move from that alveoli to the next alveoli to the next alveoli that is a neighbor to that alveoli through these same pores. So meaning air is able to move from one alveoli to another alveoli through these alveolar pores of cone. That is why they are important. That is why they are important. Okay. So this is the last thing we're going to look at today. Uh, okay, but not the least. So we're going to look at now the blood. Okay, sorry, this is blood, not good. Blood air barrier or respiratory membrane. So the blood air barrier can also be called the respiratory membrane. So there are actually three things that make up this respiratory membrane. Okay, there are actually three things that make up this respiratory membrane. So this respiratory membrane or the blood air barrier is What separates the blood from the air that is in the alveoli? The blood that is in the blood vessels and the air that is in the alveoli are separated by this blood air barrier or the respiratory membrane. So this respiratory membrane is made up of three things. So number one, we have type 1 alveoli. So take note, type 1 alveoli that lie in the alveoli are actually part of the blood air barrier, not the type 2 pneumocytes. I'm emphasizing here, not type 2 pneumocytes. This is confusing and examiners will target this. So know this. Know that type 1 alveolar cells are the ones that form the blood air barrier and not the type 2. Okay? Then the second thing is, you know that these type 1 alveoli, they form an epithelium of the alveoli. And every epithelium uh, has got a basement membrane, except for a few exceptions. One example I can give is the epithelium that is formed by the ependymal cells in the ventricular system of the brain. That one does not have a basement membrane. But this here we're talking about the alveoli lining. That epithelium has got a basement membrane. So that basement membrane uh, is actually fused to another basement membrane of endothelial cells. So endothelial cells are cells that line blood vessels. So since they form an epithelium also, they have a basement membrane. So the two basement membranes for the endothelial cells and the type 1 alveolar cells will be opposed to each other. So that means they'll be put side by side against each other. And in that process, they'll fuse together and form what we call the fused basal lamina and form what we call the fused basal lamina. Okay, so that is the end. So homework, I've not talked about two important things strategically. I've not talked about two important things strategically. So now, as your homework, go and study the storage of the larynx and the pharynx. Okay, paying attention to, for the larynx, paying attention to the cartridges that are found in there, and also the epithelial lining, its different parts. Okay. Okay, so this is the end, and thank you for paying attention. In case you want to register for physical classes or you have any queries, you can actually WhatsApp the number that is showing on the screen. Thank you so much.